Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to finish up chapter 1 for our MCAT biology playlist. We made it to objective 3, which is titled Classification and Structure of Prokaryotic Cells. Prokaryotes are the simplest forms of life, and they include all bacteria. Unlike eukaryotic cells, prokaryotic cells do not contain a nucleus or any membrane-bound organelles. Their DNA floats freely in the cytoplasm, and it's typically organized into a single circular chromosome. Now, there are 3 domains that all life can be classified into: bacteria, archaea, and eukarya. Bacteria and archaea contain prokaryotes. Both groups share certain structural similarities, like lacking a nucleus and reproducing through binary fission. But they also differ in important ways. Archaea tend to live in extreme environments, and they have unique features that more closely resemble eukaryotes than bacteria. Bacteria, on the other hand, are more widespread, and they include many species that are either harmless or beneficial to humans, as well as some that can cause disease. Actually, the relationship between bacteria and the human body is pretty complex. While some bacteria are clearly pathogenic, many others play important roles in digestion, in vitamin production, and even in immune system regulation. In fact, the human microbiome, which is a community of trillions of microbes that live in and on our bodies, that's mostly made up of bacterial species. So when we talk about bacteria, it's important to remember that not all of them are harmful. In many cases, they're vital to our health. Now, one way scientists and pathologists classify bacteria is by shape. This helps us establish a shared language for identifying and discussing bacterial species. There are 3 major bacterial shapes to know. First, you have cocci. This is spherical-shaped bacteria. Then we have bacilli. These are rod-shaped bacteria. And then we have spirilla. These are spiral-shaped bacteria. Another important classification is based on oxygen requirements. Some bacteria, called obligate aerobes, require oxygen for survival because they depend on aerobic metabolism. Others, known as obligate anaerobes, cannot survive in the presence of oxygen, and they rely entirely on anaerobic pathways to generate energy. There's also facultative anaerobes. These can survive in both oxygen-rich and oxygen-poor environments. So they're flexible, and they can switch between aerobic and anaerobic metabolism depending on what conditions allow. Finally, we have aerotolerant anaerobes. These organisms do not use oxygen for metabolism, but they aren't harmed by its presence either. So they can survive in oxygen-containing environments while still relying entirely on anaerobic processes. From here, we want to take a closer look at the structure of prokaryotic cells. As we've said before, one of the key differences between prokaryotic and eukaryotic cells is that prokaryotes do not have membrane-bound nucleus or organelles. So everything happens within the cytoplasm or at the membrane. And yet, despite their simplicity, prokaryotes are highly efficient and well adapted for survival. Let's start from the outside and let's talk about the cell envelope. This includes both the cell wall and the plasma membrane, and in many bacteria, it also includes an additional outer capsule layer for added protection. The cell wall, it provides structural support and it helps the cell maintain its shape. In bacteria, it's primarily made of a polymer called peptidoglycan. And this structure is made of sugar chains cross-linked by short peptides, and they form this rigid but flexible mesh. Now, there are two major categories of bacteria based on differences in the cell wall, and we can distinguish them using a technique called the Gram stain. In gram-positive bacteria, the cell wall is thick and it contains high amounts of peptidoglycan. These cells also have molecules that can play a role in activating the human immune system. In contrast, gram-negative bacteria have much thinner layers of peptidoglycan. What makes them unique, though, is the presence of an outer membrane outside of the cell wall. So between the inner membrane and the outer membrane is this periplasmic space, which contains enzymes and other proteins involved in nutrient processing and defense. The outer membrane also contains lipopolysaccharides, or LPS, which are large molecules that contribute to the structural integrity of that membrane, and they are recognized by the immune system. In fact, LPS is often what triggers the inflammatory response during bacterial infection. So together, the cell wall and the plasma membrane form the bacterial envelope. The envelope is not only what provides protection and structure, but it also is what mediates exchanges between the cell and its environment. Now let's talk about the flagella. These are long, whip-like structures that many bacteria use for movement. Bacteria, they don't just move randomly. They can sense chemicals in their environment, and they can move towards or away from them. And this is a behavior known as chemotaxis. Each bacterial flagellum is made up of three major parts. First is the filament. This is a long helical tail that's composed of a protein called flagellin. And this filament is connected to the basal body by a structure called the hook. The basal body acts like a tiny motor. It's embedded in the cell membrane, and it rotates to spin the flagellum and propel the bacterium forward. This rotation is powered by a flow of protons across the membrane, and it allows the bacterium to swim towards nutrients or away from toxins. And the specific structure of the flagellum varies slightly between gram-positive and gram-negative bacteria. Fun fact, archaea also have their own distinct version of flagella, but we won't be tested on that. Now, moving inward, let's talk about what happens inside the cell. In prokaryotes, the genetic material is found in a region called the nucleode. Unlike eukaryotic cells, this DNA isn't enclosed in a nuclear membrane. The DNA is usually just organized as a single circular chromosome, and it contains all of the essential genes needed for basic cell function. In addition to the main chromosome, many bacteria also carry plasmids. Plasmids, these are small circular pieces of DNA that replicate independently of the main genome. Plasmids often carry genes that could provide a survival advantage, such as antibiotic resistance, and they can be transferred between bacteria, which makes them important in both evolution and in medicine. Another key feature is the ribosomes. Even though prokaryotes don't have membrane-bound organelles, they do still synthesize proteins using ribosomes. The ribosomes in prokaryotes are structurally different from those in eukaryotes. They consist of two subunits, 30S and 50S subunits, which together form 70S ribosome. In contrast, eukaryotic ribosomes are made of 40S and 60S subunits, forming an 80S ribosome. This difference in size is important because it allows certain antibiotics to target bacterial ribosomes without affecting human cells. Finally, although prokaryotes don't have a complex cytoskeleton like eukaryotic cells, they do have a more primitive version. It's less developed, but it still helps with structural organization and division. So altogether, even though prokaryotic cells are simpler than eukaryotic ones, they're still highly efficient and incredibly diverse in structure and function. With that, we can confidently move into that fourth objective, which focuses on how prokaryotes grow, divide, and introduce genetic diversity into their populations. Prokaryotes reproduce asexually using a process called binary fission. Unlike mitosis in eukaryotes, binary fission is a simpler, more direct method of cell division. It begins when the circular chromosome of the bacterium attaches to the inner portion of the cell membrane. As the cell elongates, the chromosome replicates, and the two DNA copies move apart. Eventually, the cell divides into two genetically identical daughter cells. Although binary fission produces clones, prokaryotes have other strategies to increase genetic variability, which is especially important for adapting to changing environments. And a key part of this is the presence of plasmids. These are small circular DNA molecules that replicate independently of the bacterial chromosome. While the chromosome is essential for survival, plasmids often carry accessory genes that provide an advantage, such as antibiotic resistance. Some plasmids can also integrate into the bacterial chromosome, and when they do, they're called episomes. Prokaryotes, they use three main mechanisms of genetic recombination to acquire new genetic material. We have transformation, transduction, and conjugation. Let's begin with transformation. In this process, a bacterium takes up foreign genetic material from its environment, usually fragments of DNA from lysed cells. And if the DNA is successfully integrated into the host's genetic code, then it becomes a permanent part. And this process is particularly useful in biotechnology. It's also a natural route for bacteria to gain new traits. So to reiterate, it results from the integration of foreign genetic material into the host's genome. What about conjugation? This is often described as a bacterial form of mating. It involves the formation of a conjugation bridge between two bacterial cells, and this allows for unidirectional transfer of genetic material from a donor male to a recipient female. The donor cell, it's going to possess a specialized plasmid called the fertility factor, or the F factor. This contains the genes required to construct a sex pilus. This is a protein appendage that establishes physical contact with the recipient cell, and it forms the conjugation bridge. Once the conjugation bridge is formed, a copy of the F plasmid can be transferred to the recipient, and it converts it into an F plus cell because it has the F factor now. If the F factor is then integrated into the donor's chromosome, then the cell is referred to as an HFR strain, high frequency recombination strain. In this case, part of the donor's chromosomal DNA may also be transferred along with the F factor, and this process allows for portions of the bacterial genome to enter the recipient cell, and that increases the chances of genetic recombination. So to summarize, conjugation is the bacterial form of mating, forms a conjugation bridge, and then the male donor gives a F factor to the recipient female. Now let's cover transduction. This third method requires a viral vector, specifically a bacteriophage. So during infection, a bacteriophage can accidentally package bacterial DNA instead of its own viral genome. And then when it goes on to infect another bacterium, it injects this bacterial DNA, which may then recombine with the host's genome. This is an indirect but efficient way for bacteria to exchange genetic information. It's also worth briefly noting the existence of something called transposons. These are segments of DNA that can move from one location in the genome to another. And they're found in both prokaryotes and eukaryotes, and they can sometimes carry antibiotic resistance genes with them, and that can further contribute to genomic variation. So with that, the three methods that prokaryotes can increase genetic diversity through genetic recombination is either through transformation, transduction, and conjugation. Transformation results from the integration of foreign genetic material into the host genome. Transduction requires a vector, which is a virus that carries genetic material from one bacterium to another. And conjugation is the bacterial form of mating, forms a conjugation bridge, and that allows for the transfer of a copy of the F plasmid. Now let's take a look at how bacteria grow over time. Since they divide by binary fission, bacterial growth is often exponential, but it follows a characteristic pattern that's captured by the bacterial growth curve that you see right here. The first phase is the lag phase. So during this time, the number of cells remains constant, but they're metabolically active. They're gearing up for division by synthesizing enzymes and replicating DNA. Next comes the log phase, also called the exponential phase. Here, cells are dividing rapidly, and the population size increases exponentially. This is when bacteria are most susceptible to antibiotics that target actively dividing cells. Then, after resources begin to run low, the culture enters the stationary phase. So the rate of cell division here slows, and it eventually equals the rate of cell death. Nutrients are depleted here, and waste products begin to accumulate. Finally, we reach the death phase or decline phase. In this stage, the environment becomes too hostile, and the number of dying cells exceeds the number of new cells. And this leads to a decrease in the overall population. So although binary fission produces genetically identical offsprings, bacterial populations remain genetically dynamic due to the mechanisms like transformation, conjugation, and transduction, as well as the activity of transposons. And the population growth curve follows a couple of phases: lag phase, log phase, stationary phase, and finally death phase. So that's putting that all together so that we can move into our last and final objective in this chapter. Here we're going to talk about viruses and subviral particles. Viruses are a unique class of biological entities. They contain genetic material, either DNA or RNA, and they're surrounded by a protein code called the capsid. So this capsid is also enclosed by a lipid-containing envelope, which is typically derived from the host cell's membrane. However, unlike living cells, because viruses are not living, viruses are acellular, and they cannot survive or replicate on their own. So that means they require a host cell to reproduce, which is why they're considered obligate intracellular parasites. One specific type of virus is the bacteriophage, which infects bacteria. And bacteriophages have a specialized structure that includes a tail sheath and tail fibers. The tail sheath acts like a syringe to inject genetic material, and the tail fibers help the virus recognize and bind to specific bacterial hosts. Now, when we look at the genome of a virus, it can take several forms. Viral genomes can be made of either DNA or RNA, and they may be single-stranded or double-stranded. In the case of single-stranded RNA viruses, the RNA can be classified as either positive sense or negative sense. Positive sense RNA can be directly translated by the host's ribosomes, while negative sense RNA needs to be converted into a complementary strand first. Another important group of viruses are retroviruses. They're interesting because they contain a single-stranded RNA genome, but unlike typical RNA viruses, they use an enzyme called reverse transcriptase to synthesize a complementary DNA strand from their RNA. And this DNA is then integrated into the host genome, and that allows the virus to replicate along with the host cell's DNA. Now that we've discussed viral structure and genome types, let's take a closer look at how viruses replicate. The viral life cycle typically follows a sequence of steps: attachment, penetration, biosynthesis, assembly, and release. The cycle begins when a virus attaches to a specific receptor on the surface of a host cell. And this receptor recognition is highly specific, and it determines which hosts and cell types a virus can affect. Now, once attachment occurs, the virus enters the host cell either by membrane fusion or endocytosis, depending on the type of virus and whether it has an envelope. Once inside the host, the viral genetic material is then released into the cytoplasm. Then, the host cell's own enzymes and ribosomes are hijacked to replicate the viral genome and synthesize viral proteins. This stage is known as biosynthesis. The viral components are then assembled into new virons in a step that's called assembly. Finally, the mature virons are released from the host cell, and this can occur through lysis, which destroys the host cell, or through extrusion, which allows the virus to exit while keeping the host cell alive. Lytic release is more common in non-enveloped viruses, whereas extrusion or budding is typical for enveloped viruses. Some viruses, especially bacteriophages, those viruses that infect bacteria, they can enter an alternative cycle called the lysogenic cycle. Whether a bacteriophage enters the lytic or lysogenic cycle depends on environmental conditions and host cell status. In the lytic cycle, the virus immediately begins replicating upon infection with very little regard for the survival of the host cell. It produces large numbers of virons, and it ultimately causes the host cell to lyse and release these new viral particles. In contrast, during the lysogenic cycle, the viral genome integrates into the host's DNA, and it becomes a provirus. And in this dormant state, the viral genetic material is replicated along with the host's genome each time the cell divides. And this virus may remain in this latent form for extended periods of time until a trigger causes it to exit the lysogenic cycle and enter the lytic cycle. Now, we're going to finish this chapter up by briefly discussing two unique infectious agents that fall outside the standard definition of a virus. Those are prions and viroids. These are classified as subviral particles because they are even simpler than viruses, but they can still cause disease under specific circumstances. Prions are infectious proteins that do not contain any genetic material. What makes them dangerous is their ability to induce the misfolding of normal proteins in the host, particularly in neural tissue. And this misfolding disrupts normal protein structure and function, and that can lead to progressive neurodegenerative diseases. Since prions are made entirely of protein and they lack RNA or DNA, they challenge our traditional understanding of what constitutes an infectious agent. Viroids, on the other hand, are composed solely of a short strand of circular, single-stranded RNA. And unlike viruses, they don't encode proteins and they don't have a protein coat. Instead, viroids exert their effects by interfering with gene expression in the host cell. They're known to infect plants, and they can disrupt normal growth and development, which can cause some damage. So to summarize, viruses are infectious agents with genetic material, either DNA or RNA, enclosed in a protein coat and often with a lipid envelope. Viroids are tiny plant pathogens. They're made only of RNA. And prions are misfolded proteins capable of spreading their misfolded shape to normal proteins. And with that, we have covered everything we need for chapter 1. I hope it was helpful. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.